Good afternoon, everyone. I hope that you guys are having a good day. Sorry for my delay and scrambling to get things going here. Um, at any rate, I hope that you guys are doing all right. I hope you've had a chance to look over your exam, whether it was in person or the online version. Um, those are all available. I posted an announcement last night. I think maybe it was this morning. I can't remember. A lot's happened. Um, Detailing some things, some changes about the week. For example, Thursday is Juneteenth, which is a federal holiday, so we will not be meeting for our class on Thursday. Um, I don't want you. I had it on the calendar. I just slipped my mind. I apologize for that. Um, but I don't want you to think like, oh, great, he had it scheduled for that, and now he's just gonna, overload us with a bunch of other stuff. I'm not going to do that, and I'm not going to be like, ooh, I missed a day, and make it up on this day. Um, Nothing will be assigned or due on the 19th. I believe I just made the change to your quiz, which was scheduled for the 19th. I moved that to Friday the 20th. Um, so that will be available starting at 12 p.m. on Friday and will close at 11:59 p.m. We will have class on Friday the 20th as scheduled or as we've had on the calendar. Um, and then your homework or your problem set is going to be due on Sunday. Um, I have to make the problem set questions available in just a little bit, and I'm going to do that shortly after our class. And that's pretty much the primary important things for this week. Um, we are going to be tackling glycolysis this week. I believe that I have all of the PowerPoints for this week up on our Canvas site, so you're welcome to, get after those as soon as you want. Um, then I also have started the writing the practice exam. So I've got a couple of questions for that ready to go. Um, so you can look at that. You can start working through those as soon as you're ready. But in the meantime, I wanted to go ahead and tackle kind of our last little bits of inhibition. And then we're going to jump into glycolysis. We've got glycolysis scheduled for today and tomorrow. Then we'll do lipids and membranes on Friday. If we get a little bit ahead on glycolysis or make it through more than I expect for today, then we will go ahead and move on to lipids and membranes tomorrow so that we can kind of move through things as briskly as possible. So this right here is a slide that we've already seen. This is just a little bit of context and a little bit of background. So the PowerPoint over enzymes, the last like maybe it was as many as like 12 or 13 slides. That's where we're taking off. So enzyme inhibition. Enzyme inhibition, whenever I think of it personally, I always think of like drug design and cancer therapies. This drug has been designed in order to block the activity of this particular enzyme. Um, whether that's an enzyme in central metabolism or something that controls the cycle in which cells are replicated, some fundamental process. That's kind of what I always think about personally. We discussed the two major kind of frames or possible ways that an enzyme can be inhibited, and it can be inhibited in a irreversible fashion or a reversible fashion. An irreversible fashion simply means that that enzyme is basically blocked from ever functioning again. Um, these are sometimes, or these types of inhibitors are sometimes referred to as inactivators because that's what they do. They take that polypeptide and they basically make it completely useless for forever. Um, they can bind permanently to the enzyme, but other things that can be done is that they can chemically modify an amino acid. So making some sort of chemical modification that cannot be kind of naturally undone. And that's not just any random amino acid. That would instead be targeting an amino acid within the active site of the amino acid of the enzyme. Now, reversible enzyme inhibition is the type of inhibition that I would argue is more commonly discussed and studied when it comes to a biochemistry class, because this is like it's more transient, it's more temporary, um, and this is where we have some simple models of inhibition. So if ever you've opened up an MCAT study book or something like that and it asked about enzyme inhibition, chances are it's asking about this type of inhibition. The reason for that is it's more interesting because irreversible inhibition, it knocks out the enzyme's function. Done. That's it. It doesn't necessarily like make it something that you can alter. Um, so reversible enzyme inhibition. We talked about one specific model, and the model that we talked about was competitive. A competitive inhibitor, and I'm going to try to use language that will make some sense to you guys in terms of what I'm talking about and giving you guys the context. Chances are it looks like this is a little bit of an aside. It looks like there's two boxes that are blocking out portions of the screen. Um, and I want you guys to do your best to kind of ignore those. That one of those is my chat window, and then the other one is my window where I can see anyone's face. If you have your camera on, I will do my best to rearrange those to make sure that you guys don't lose sight of anything that's important on the screen. So right now they're not blocking out anything important on the screen. Okay, so competitive inhibition, that's the type of inhibition that we previously talked about. And with competitive inhibition, you basically have an active site for your enzyme that is going to bind your inhibitor or your substrate, the S or the I. It's not going to bind both of them simultaneously. It's going to bind one or the other. And in competitive inhibition, there's a couple of things that can happen. With a competitive inhibitor, your enzyme represented by E can bind I, the inhibitor. And if it does bind I, well, then what's going to happen is you're going to form this EI complex. Now, in an EI complex, there's no room for your substrate. Alternatively, if the substrate binds to the enzyme, well, you're going to form this ES complex. There's no room for the inhibitor. So with a competitive inhibitor, it's kind of one or the other. Your enzyme is either going to bind the substrate or the inhibitor. If it binds the substrate, great, you're going to make the product. If it binds the inhibitor, sorry, you're not going to make the product from that. Now, the key part about this is that just as you bind the inhibitor, you can dissociate, you can lose the inhibitor. And there is basically a constant related to that, represented as the Ki. So basically, a Ki is similar to a Km, but it's a measure of affinity that your enzyme has for the inhibitor as opposed to the substrate. Now, the same sort of idea goes, like the lower the Ki, the stronger the affinity that your enzyme has for that inhibitor. Now, when we look at competitive inhibition, we've already talked about this. I just want to go back over it. A couple of things can change. The Vmax is going to remain the same. As the Vmax, so as soon as your enzyme binds to the substrate, you're going to make the product. If your enzyme binds to the inhibitor, you're not going to make the product. Now, what that essentially means is you've got to stack the deck in favor of your enzyme binding your substrate as opposed to the inhibitor. So if your enzyme says, hey, I can either bind to the substrate or the inhibitor, and you want your enzyme to make a product, give your enzyme a ton of the substrate. Because if it's given one or the other, or it's given equal concentration of each of them, well, it's going to kind of say, I'll flip a coin. Now, whenever it binds to that substrate, there's nothing slowing that process down. As soon as you form ES, you can make the product. So that's why your Vmax does not change. And we see that represented in a Lineweaver-Burk plot right here, our LB plot, and then our Michaelis-Menten plot. These lines right here, these, I guess it's kind of like a purple, blue, and an olive colored lines on an x-axis that goes on into infinity. These all kind of plateau together. They merge together because the Vmax is effectively going to be the same. Now, the KMs, on the other hand, those are what are going to change. And that's known as your KM apparent. So that's how you can compare the KM in the presence of the inhibitor or in the absence of an inhibitor. Now, when it comes to kind of visualizing that, well, this is your enzyme right here. It's not happening. No, it's about to. Nope, it's not happening. I have a sneeze that's right here. I know it, but it's not happening for me right now. Here's our enzyme in both cases. The active site of this enzyme, it's very similar. An inhibitor has a complementary region to that active site, just as the natural substrate has a complementary region to that active site. And here is our reaction taking place. Oh, we bind the substrate, we make the product. Great. Alternatively, if we bind the inhibitor, well, then we kind of lock this whole system up. And then what happens when our substrate comes along? It says, oh, rats, I missed it. Okay, bye-bye. Now, in terms of what does the substrate or the, like, if you think about this from a drug design perspective, I think it's important to think about, okay, well, if you're interested in designing drugs, designing inhibitors, then your interest in your area of interest should be organic chemistry and kind of the interface of organic chemistry and biochemistry, because you need to know a lot about an enzyme and you need to that enzyme's active site, like what are the amino acids within that active site, and then how would you design a drug to actually get delivered to that and interact with that. So if you think about, let's say, an active site, and let's say that within an active site of an enzyme, we have the following amino acids: arginine, serine, and let's say for the heck of it, we've got a leucine and a glutamic acid. Arginine, serine, glutamic acid, those three residues, they have those have some interesting interactions. Okay, we've got an arginine that's probably going to be positively charged, a glutamic acid that's probably going to be negatively charged, and a serine that participates in hydrogen bonding. If those are the amino acids that make up the active site, do we want to design an inhibitor that is simply hydrocarbons? Add to the chat a number if you say yes and a letter if you say no. Absolutely. We do not want to design an inhibitor that's just a bunch of hydrocarbons, or that's a alkyl or an aliphatic chain. Because what type of interaction is this going to elicit? This is going to have hydrophobic interactions, or that's going to kind of require hydrophobic. And that's not what this needs. We need something that has charged species on it. So then we design something that kind of complements that. Now, whenever it comes to other types of inhibitors, there are some examples of competitive inhibition. For example, methanol poisoning. that is treated with ethanol. Why is that? Well, let's look at the similarities of these molecules. Methanol, ethanol. The difference between them is simply a methyl group. Now, alcohol dehydrogenase, ADH, the enzyme that if you um, so that's why some people get kind of blushed red cheeks whenever they drink booze or drink alcohol. They kind of have like a flushed look because alcohol dehydrogenase is very active. Converts methanol to formaldehyde. Formaldehyde is going to be very toxic. That can cause some pretty intense liver damage and or brain damage. It can cause blindness and death as well. Ethanol competes with methanol for binding to alcohol dehydrogenase. So in the event that somebody accidentally drinks methanol, well then sounds strange, but give them a lot of alcohol, give them a lot of ethanol, and that will basically compete it away. So they're going to be competing for the same enzyme. I'm sorry, just 1 second. I have to blow my nose. So that's an instance where a physician might say, "Oh well, much alcohol," or maybe it's not. I don't know how that's actually administered. Uh, so don't listen to my advice on that. Not a physician, doctor of philosophy. That's what I am. Okay, here we go. Now, another type of inhibitor. This is one that is inherently kind of confusing because one of the other inhibitors we'll learn about has a very similar name, an uncompetitive inhibitor. I always kind of think of this type of inhibitor as the opposite of a competitive inhibitor. The reason I say that is because a uncompetitive inhibitor, which I'm going to circle that I here, what an uncompetitive inhibitor will not do is it will not bind the free enzyme. It will not compete with the naturally occurring substrate. So it's opting out of competition. So it will not bind to the natural or to the free enzyme. And by not binding to the free enzyme, what instead it will do is it will bind to the enzyme substrate complex, or this ES. So here's our model of our free enzyme, our substrate. So in the presence of an uncompetitive inhibitor, our substrate will bind. Okay, so here's our enzyme, here's our substrate. That complex will form. The free inhibitor will only bind to that complex. So you can kind of think about this as maybe like the geography or the space that exists for the inhibitor to bind, or for an uncompetitive inhibitor to bind, will only be there after the substrate has actually bound to it. So you could kind of think about this as, a little bit similar to an ordered ternary mechanism. In the sense that, oh, this is an ordered system, this is a process. So an uncompetitive mechanism or uncompetitive inhibition, I would think about it very similar to this, because an uncompetitive inhibitor will not bind before the free substrate binds. Now, what that leads to is here's our Lineweaver-Burke plot. Here is our x-intercept, here's our y-intercept. Our y-intercept tells us our 1 over v max, and then our x-intercept is our negative 1 over km. Okay. Well, answer in the chat with a Y or an N. A Y for yes, an N for no. Does the Vmax change in the presence of an uncompetitive inhibitor? Yes, it absolutely does. Okay. Does our Km change? Does our enzyme's affinity for our substrate change in the presence of it? Perfect. Now, an uncompetitive inhibitor will cause Vmax and Km to change. Now, this is going to seem a little bit odd, but what this is going to do is it's going to cause this is here, let me circle some points. Here's our initial Vmax, and here's our 1 over Vmax apparent. So I think everyone agrees that our Vmax will change. Done. But will our Vmax increase or will it decrease? This is a point where people get a little bit stumped because if you look at this point and this point, you know, I'll go with the point where the X is, compare that to the point where the Y is. If you look at that on a on this Y axis, moving from point X to point Y, I think everyone agrees that that number increases. We could look at this and say this is approximately 0.0025. The similar point is, I'm going to call that 0.0035. Okay. So this is with the inhibitor. That number does go up. But does the Vmax go up? Answer that in the chat with a Y for yes or N for no. Does the Vmax? No, it does not. And that's always something that I've had people kind of they struggle to wrap their mind around. And the reason that is because the nature of this y-axis or the values on this y-axis, that's a reciprocal value. So those reciprocal values, if they go up, that means what's in the denominator, which happens to be Vmax, is going down. So that's an important point to kind of keep straight. I think it's also just kind of helpful to remember what these graphs look like. And so when you see a question on the test that's as general as what does an uncompetitive inhibitor do to the Vmax? Then say, okay, well, I know how to draw that plot out. Okay, here's this, and then just throw in some dummy values. And whenever I say dummy values, I basically mean 1, 2, 3, 4. Don't mess with decimals. And then if you really want something very precise, say, okay, this is 32 or 3.2, and this is 2.1. Done. Decimals are going to throw people off for forever. But this way you don't have to mess with that. Okay. The same thing is true for the KM. So here's another layer of the complexity. What does an uncompetitive inhibitor do to an enzyme's affinity for its substrate? So E affinity for substrate. How does, in the presence of an uncompetitive inhibitor, how does my enzyme's affinity for my substrate change? Does it go up or does it go down? U for up and D for down. Now, I got one response for that, and that one response was right. And that's pretty standard to get one response for that because most of the time when people look at that, they think to themselves, okay, my enzyme's affinity for the substrate. Okay, if my affinity goes up, that's good. If my affinity goes down, that's bad. An inhibitor that causes my enzyme's affinity for the substrate to go up, wait, that doesn't comport. That sounds like that's a generally good thing because wouldn't you want your enzyme to be able to grab that substrate out of a complex mixture? Yeah, you would. Well, in an uncompetitive inhibitor, your enzyme, or in the presence of an uncompetitive inhibitor, your enzyme's affinity for the substrate does go up. The Km goes down. We already know that those two things are inversely related, so that kind of makes sense. But one of the things that I always try to think about with respect to this model up here, and I'm going to scribble some of this out. Okay, I'm going to scribble this out. Now, what I want you to look at is this right here. That right there, those letters, E plus S yields ES, yields E plus P, and ES binds I to go to ESI. So let's say, when we're doing enzyme studies like this, one of the things that we keep constant is we keep E concentration remains constant. Can I get in the chat a Y if you agree with that statement? Or an N if you disagree with that? Okay. So our enzyme concentration from trial to trial to trial is going to remain constant. That's great. Now, let's say that we design an experiment with an uncompetitive inhibitor, and we put 100 copies of our enzyme into this mixture. Now, we're going to let this reaction go for, let's say, 5 minutes or something like that. And so whenever we get to this point right here where we've concluded kind of one round, we've got our free enzyme again. What's that free enzyme going to do? What will this enzyme do? I'll give you a second to add to the chat what that enzyme is going to do. It's kind of reset. It's ready to go again. It's going to bind to the substrate. You're absolutely right. So you can kind of think about this as the enzyme goes through this cycle. Can I get a Y in the chat if you follow that? Yeah, I get it. It goes through a cycle. And it's going to continue doing that. It's going to do multiple cycles per 10 minutes. Would everyone agree with that? Add a Y to the chat if you agree with that. Okay. So now that we've agreed on those important principles, let's say that we've got 100 copies of our enzyme. We have no shortage of our substrate. Arguably, whenever you're doing enzyme studies, you've got, let's say, I'll just give a nice round number. We've got 1,000 copies of the substrate. And, in round number 1, right here, let's say that let's say just for the heck of it, we've got 500 copies of the inhibitor. So we're going to end up with 100 of this ES complex. Could everyone add to the chat that, yes, I agree with that? Add a Y or an N if you don't agree. Okay. So we have no issue with all of our enzyme forming this ES complex. And we got 500 copies of this inhibitor. And let's say that, the uncompetitive inhibitor is not something that's super enticing. But some of that ES complex is going to be diverted. And let's say of those 100 ES complexes, about 20 of them bind the inhibitor and end up here. 20 of them end up as ESI. So if 20 of them kind of get compartmentalized or kind of siphoned off as ESI, how many E do we end up with here? Okay. So we're going to lose some. Would everyone agree with that? For every successive round, we're going to lose some of them. Now, what that means is that we're not doing this just one round. We're not doing it 2 rounds. Let's say that we do 10 rounds of this. At some point, we're going to reach an equilibrium where, maybe we end up with like 45 ESI. And then we got 55 of the free enzyme that's cycling through. And yeah, this complex falls apart, but then it reforms. What that means is that even though we designed the experiment where we kept our enzyme concentration constant, we did not change it. What did this uncompetitive inhibitor do? Our uncompetitive inhibitor kind of artificially drove down our enzyme concentration. So we didn't, set it up with any less, but it just gets siphoned off, which results in our affinity, our enzyme's affinity for our substrate to go up. Because there's less of that enzyme there, it's going to bind to that substrate more easily and readily. Can I get a Y if that makes sense, or a no, or anything else? A Y is I'm good. Any other letter, any other character is I don't follow. Okay. The important takeaway from this is that Vmax goes down, Km goes down, but Km is an inverse number, so the lower Km means higher affinity. So when I'm comparing a competitive inhibitor from an uncompetitive inhibitor, I'm thinking competitive inhibitor does nothing to Vmax. Or sorry, does nothing to Km. Nope, that's wrong. A competitive inhibitor does nothing to Vmax. Competitive does not change Vmax. Km. Km goes up in the presence of a competitive inhibitor. An uncompetitive inhibitor causes both Km and Vmax to change. Vmax goes down, Km also goes down. Now, when you look at these two lines on an uncompetitive inhibitor plot, if you were to do any studies with an uncompetitive inhibitor, these lines right here and here, this one, this dashed line is the one that's with an inhibitor present, and this solid line is without an inhibitor present. Those two lines, what you're going to see is that they run parallel to one another. And that is what you will see. That is the trend that you will see. So anytime you talk about inhibition and you say, oh, well, the lines are not intersecting, well, they are either going to intersect at some point or they run parallel to one another. When they run parallel to one another, that is the hallmark of an uncompetitive inhibitor. An uncompetitive inhibitor causes those two lines to run parallel to one another, which means that the Km and the Vmax change at basically the same relative rate. Now, that's what you're going to see on the Lineweaver-Burke plot. That ESI complex, which I want to share with you guys, one of the things that I always like to ask about is which complexes form. The EI complex will not form in the presence of an uncompetitive inhibitor. The ESI complex will not form in the presence of a competitive inhibitor. So I think it's helpful to compare and contrast those two to one another. Now, sometimes it's referred to as a third, and sometimes it's referred to as a third and fourth type of inhibition. And this is commonly known as mixed inhibition. The reason for that is that a mixed inhibitor is kind of capable of acting like a competitive inhibitor. It's also capable of acting like an uncompetitive inhibitor. What you see here on the right-hand side of our screen, right there, is pretty accurate. A mixed inhibitor can bind to the free enzyme and form the EI complex. Alternatively, a mixed inhibitor could bind to the ES complex, forming the ESI complex. So if EI forms, well, that's very similar to a competitive inhibitor. If ES forms, well, that's not the case of a competitive inhibitor, but that's what an uncompetitive inhibitor does. So the way that I always like to ask questions about this is, hey, this type of inhibitor causes EI to form, also allows for ES and ESI to form, in which case that is a mixed inhibitor. Now, some of the things about the Lineweaver-Burke plot when it comes to a mixed inhibitor is you always like to ask, where are the two lines, the no inhibitor line and the plus inhibitor line? Where are they intersecting? Now, I like to do this just because it helps me kind of keep things straight. I'm going to draw a dashed line to represent no inhibitor. And what I want you to do is I'm going to draw, so a solid line for no inhibitor, and now I'm going to draw a dashed line to indicate plus inhibitor. Based on this dashed line, what type of an inhibitor is shown right here? Tell me in the chat. And I'm moving my stuff so that I'm not blocking. Oh, no, that just blocks it all. What type of inhibitor is shown there? Perfect. A competitive inhibitor. All right. Now, the way that I always like to think about this is, how do the lines intersect, or where do they intersect? This is a y-axis intersection. Okay. Now, with an uncompetitive inhibitor, it's going to look like this. No intersection. Let me change that. No intersection. A mixed inhibitor is a little bit kind of tricky. Here what we see, if you remember back to, I remember learning it in third grade, quadrants on an XY axis. You have quadrant 1, 2, 3, and 4. Well, if your lines intersect in quadrant number 2, that's an indication of a mixed inhibitor. So kind of pinpointing where these lines intersect is really helpful to identify what type of inhibitor that you're looking at. In addition to that, it helps you also look at, well, which variables change? Is my Km changing? Is my Vmax changing? Now, when it comes to a mixed inhibitor, there are some levels of it that are a little bit more subtle. So if we look right here, we also, under the umbrella of mixed inhibition, we have non-competitive inhibition. This is confusing when you have an uncompetitive inhibitor. A non-competitive inhibitor and an uncompetitive inhibitor are not the same thing. In the case of a non-competitive inhibitor, you can have this whole thing take place. You can have your EI form, you can have IES form or ESI form. And what you're going to see with a non-competitive inhibitor is you're going to see an intersection of your your no inhibitor and plus inhibitor intersect on the x-axis. So think about this as, if your lines intersect on the y-axis, that's a competitive inhibitor. If they intersect on the x-axis, that is a non-competitive inhibitor. Quadrant number 2, that is a mixed inhibitor. And then, if they don't intersect at all, that would be an uncompetitive. So intersection, where is it? If it's on the y-axis, competitive, x-axis is non-competitive, quadrant 2 is mixed, and no intersection is uncompetitive. That table right there is going to be very helpful. Well, it's not really a formal table, but make that into a formal table, and that's very valuable information. That sums all the different types of inhibition up. Now, this is essentially all of those graphs. If we look right here in the top left, our intersection is on the y-axis. This is competitive. So I'm going to rewrite. Now, I'm going to go over here in the lower right-hand corner. That intersection of our inhibitor and no inhibitor is on our x-axis. That is non-competitive. Above that, well, we have no intersection. That would be uncompetitive. And then over here, we've got quadrant 1, quadrant 2. That intersection right there is an indication of purely mixed. Okay. Now, just so you guys know, I like to show pictures of these graphs. And I like to ask the question, what type of inhibitor is illustrated here? But I always like to throw it in a kind of scenario-based question. And what I mean by that is I'll say like a new drug has been designed and it has been challenged against this enzyme. Here are the results. What type of inhibition is shown? Then, to add another layer to that, I like to ask the question, well, what complexes are forming? Is EI, is EIS, is ES? What complexes are present in this type of inhibition? And if you can know that, that's going to be very helpful. Alternatively, what I like to ask are, well, what's happening to your Vmax? What's happening to your Km? Does your enzyme's affinity for the substrate change? Does it go up? Does it go down? And whenever I show you these graphs, you guys can use scratch paper on your exams. Any one of these graphs. If you're like, oh, I don't know I can't remember what type of inhibitor it is, but he gave us this graph. You know what I'm going to do? I'm going to say this is -1, this is -2, this is -3. What's happening to my Km? Okay, well, -3 equals -1 over Km. That is for no inhibitor. Okay, so that's Km equals 0.33. Then, okay, over here, -2 equals -1 over Km. Km equals 0.5. So now I've got this one right here and this one right here. Okay, cool. That tells me my Km has gone up. Okay, so if my Km goes up, what does that mean for my affinity? Oh, that means that my affinity, oopsie, substrate affinity goes down because my Km goes up. If you can do those sorts of calculations, that will help you out a whole lot to kind of like critically think and carefully work through these questions. Okay, so there we go there. And I think that this isn't the last slide, but it's close to it. Here's just kind of a summary of the different types of inhibition and the effect of the inhibitor. A competitor, so with no inhibition, you have no change. In the case of a competitive inhibitor, it causes your Km to go up. Uncompetitive causes your Km and your Vmax to both go down. In mixed inhibition, what that is showing is that's showing all of the possible outcomes given these different graphs down here. Your Vmax, oopsies, your Vmax goes down, but your Km may increase or decrease or not do anything. if you know these four graphs and can interpret them, that's going to help you out a whole lot in answering questions about inhibition. Okay. Does anyone have any questions about models of inhibition? Um, I'll give you a couple of seconds to put them together, or I can hang around for a minute after class. And
I have a quick question.
go for it.
Does non-competitive, in its like in the enzyme, like the equilibrium, like little picture that you showed, does it differ significantly from like the ES-ESI complexes of a mixed?
Say
That
that
makes
one more
sense.
time. I'm sorry.
Does like the E plus S ES like um, complexes and the way it binds inhibitors, does that differ significantly in the non-competitive?
I couldn't really. I'm having trouble understanding, and I'm just gonna. It sounded to me like you were asking about the makeup, the ES, the ESI complexes in the presence of a non-competitive inhibitor. Is that correct?
Yes, specifically versus the mixed.
Okay. So in the case of a mixed inhibitor versus a non-competitive inhibitor, what you're going to see more times than not in the presence of a non-competitive inhibitor is you are going to see. Okay, so here I brought it back to the non-competitive inhibition. More than likely, what you're going to see in the case of a non-competitive inhibitor is this. More than likely, you're going to see ES form and the IES or the ESI form. Now, what that means effectively is that a non-competitive inhibitor, chances are it can bind to the same site or the same active site as the natural substrate does, but it will be displaced. Like, think about it as the affinity that the enzyme has for that inhibitor over the natural substrate is really low. So it's like I'm trying to think of an analogy. I can't think of an analogy right now. But the inhibitor and the natural substrate have the same landing spot. But the difference between them is that the inhibitor has another landing spot elsewhere on that same enzyme. So that's why it can kind of find its place right here um, and form that IES complex. Now, if you were studying, if you put the free enzyme and that non-competitive inhibitor in the same location, well, you would form EI. But as soon as you put that substrate in, boom, you're going to swing right back over to the ES complex. Does that answer your question? Did okay. Okay, I hope that answers. Yeah, so if that didn't answer it, please feel free to add to the chat. Miracle, did you have a question?
Yes, I just wanted to find out if we're gonna need to know those equations on the,
The
should I say,
equations for the line?
yes, these ones.
No, it's not a very high priority to me. Because I'm not going to ask you to solve for what's the V sub zero. And honestly, all of this is these are just the equations for the line that show like how it all tracks. And after you've plotted it all. But I'm not going to ask questions explicitly about those.
Okay. And my second question was that you mentioned that you could ask uh this inhibitor or this type of inhibitor forms this complex. And so with two different types of inhibitors that form the same, how do I explain it? Like for example, non-competitive can form EI and so can mixed, I think. So will there be like a clue in the question to like differentiate the two?
Yes, so that's a great question. And the way that I would that's those are the sorts of questions where there's going to be kind of nuance and subtle subtle clues within the question, like the wording. For example, if I were to distinguish between non-competitive and, or if I were to discern between mixed or competitive and uncompetitive, those are a little bit easier. Because it would be like in beaker A, you see E, ES, EI, EIS. In beaker B, you see E, ES, ESI. What type of inhibitor is in beaker B? And the comparison between mixed and non-competitive would be a little bit more subtle, and it would be something like I would I would try to quantify it, and I would say something like 100 copies of the enzyme are added to a beaker, an infinite amount of the substrate is present, and a working concentration of the inhibitor is present. From the 100 copies of the enzyme, you see 5 free copies of the enzyme. You see 56 copies of the ES complex. You see 1 EI, and the remainder is an IES. What type of inhibitor is this? Or let me rephrase that. This inhibitor is behaving as which type of inhibitor? Is it a competitive, uncompetitive, non-competitive, or mixed? And that would be non-competitive because I would be saying, yeah, it does form, but compared to everything else, it's pretty negligible. But chances are, my when it comes to comparing the different types of inhibitors, I always like to stress like the the stark differences between competitive and uncompetitive, and then mixed and the other two. Okay,
Thank you.
absolutely. Great question. Okay, did I have another question? Okay. So now we're going to switch gears and we're going to talk about glycolysis. And we're using glycolysis kind of as I would say, a kind of a model for introducing different classes of enzymes. Um, one of the pre-recorded lectures that I gave you guys, or set of lectures, was all about sugars. And when it comes to glycolysis, what we are focused on is glucose, and we are focused on the ringed form of glucose. Now, there are parts of this that are very that okay. Glycolysis is basically your most central metabolic pathway. Um, Historically, whenever anyone takes a metabolism class, glycolysis is a pathway that they're introduced to first. And the reason for that is because it's all about extracting ATP from glucose. So getting energy from glucose. Now, the learning objectives for this are I want you to be able to describe the overall purpose of this. I want you to um, understand that the purpose or the role of glycolysis is to make ATP and NADH. Now, ATP, I think if I had to guess, it was probably in like 6th grade in Mrs. Guin's science class that I learned about ATP, or I first heard it. And, we were talking about like a model of a cell. Here is a cell, here's the mitochondria. The mitochondria is the powerhouse of the cell, generates a lot of ATP. Okay, cool. Um, I would say that ATP is kind of like, trying to think of an analogy. Okay, ATP is Kim Kardashian and all of her sisters. And NADH is Kris Jenner. Kris Jenner is the one behind the scenes. She's the marionette. She's pulling the strings to make all of this happen. Um, and I mean, Kris Jenner has her own like public persona and profile, um, but it's almost like they're the unsung hero. NADH is kind of the unsung hero. Like you hear a lot about ATP, but what you need to hear about is NADH because NADH is a high energy compound that from which you can generate lots of ATP. So we're going to talk about those two molecules. I want you to be able to understand glycolysis in two different phases. Those phases are the investment and payoff phase. And I will do my best to use that terminology, but I sometimes will say investment and return. They're synonyms for one another. I want you to be able to summarize the net reaction of glycolysis, including the reactants, products, and the energy that's produced. I want you to recognize the three irreversible steps of glycolysis. Those are, spoiler alert, hexokinase or glucokinase, phosphofructokinase 1, also known as PFK1, and pyruvate kinase. I want you to know a little bit about their role in regulation. I want you to be able to describe the role of hexokinase and glucokinase in glucose phosphorylation and how their activity differs in different tissues. Then, last two items, I want you to understand the role of NAD plus in glycolysis and how it is regenerated in aerobic versus anaerobic conditions. I want you to be able to classify the enzymes of glycolysis using the EC numbering scheme. Okay, so if you're thinking to yourself, do I have to know the steps of glycolysis? It's going to help you. I'm going to give you a figure that I think would be very beneficial to you. There will be some questions on your exam that will give the full names, but what lots of people like to do is they like to go with, can I know the initials for that enzyme? And if you can convert the initials into the full name, then you're going to be golden. Now, glycolysis as a whole, we are taking glucose, this compound right here. This is our 6-carbon molecule. This is glucopyranose. This is alpha glucopyranose. This 6-carbon molecule is going to be split into pyruvate, which go ahead and somebody tell me in the chat, how many carbons does pyruvate have? 3. Perfect. So if you're going to break a 6-carbon molecule into a 3-carbon molecule, how many carbons are left over? Yeah, 3. So I think that it's appropriate and makes sense that we're going to take one 6-carbon molecule and break it into 2 identical 3-carbon molecules. That molecule is known as pyruvate. Pyruvate is a molecule that people struggled to draw, and there's really no reason why you need to struggle to draw it. It is essentially, well, a 3-carbon chain. 2 of them are carbonyls. One of them is a carboxylic acid and a methyl group. If you can remember that, that's going to help you out a whole lot. That's going to speed up things. In order to do that, we're going to do an investment of ATP, PI, or inorganic phosphate, ADP, which is adenosine diphosphate, and NAD+. NAD+ is a nucleotide. NAD+ stands for nicotinamide adenine dinucleotide. We are going to recover ATP and NADH. Now, could anyone add to the chat, what is the fate of pyruvate? The fate. There's multiple fates of pyruvate. Can anyone tell me one of the fates of pyruvate? Yeah, it's going to be broken all the way down to carbon dioxide. How we're going to do that is we're going to go through the Krebs cycle, or the TCA, the tricarboxylic acid cycle. Another fate is that it's going to be in your muscles. Does anyone know what happens to pyruvate under intense, rigorous exercise, or if you go for a run? What's going to happen to it? It's going to lead to the formation of lactic acid. So lactate is going to accumulate in your muscles, and then you're going to get crampy, and then you're going to get angry, and you're going to say, "I never should have gone on this run anyway. I knew it." Ugh. But those are two of the fates. Um, Outside of human beings, yeast and plants, not many plants, but some plants under certain conditions will do what's called ethanolic fermentation. And they will take that pyruvate and they will make ethanol. Um, so those are your three fates of pyruvate: tricarboxylic acid cycle, lactate, or ethanolic fermentation. Sometimes you'll hear lactate as lactic acid fermentation, but that's kind of the name of the game. So that is what we're trying to do. We're trying to get 6 carbons and split them into 2 identical pyruvate molecules. Now, our overall view of glycolysis is shown here. I mentioned the initials or the acronyms that people like to use. I'll give you a little bit of a sense of this. What we have on the far right-hand side, we've got our molecules. Glucose, I can draw a star better than that. Can't I? Yeah, I can. Glucose is converted to glucose 6-phosphate, which is converted to fructose 6-phosphate, which is converted to fructose bisphosphate, fructose 1,6-bisphosphate. That gets split into glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. Those are all of the compounds in glycolysis that are formed. The enzymes that do this are HK or hexokinase, PGI or phosphoglucose isomerase, PFK1, sometimes just known as PFK or phosphofructokinase 1, aldolase, GAPDH or glyceraldehyde 3-phosphate dehydrogenase, and triose phosphate isomerase, also known as TIM, phosphoglycerate kinase PGK, and phosphoglycerate mutase or PGM, enolase, and last but not least, pyruvate kinase or PK. Now, I have not, that's this figure right here in a nutshell is glycolysis. It has everything. It has all the steps where ATP is generated, where ATP is invested. We're going to dig into each one of those, and I'm going to give you a little bit of time on each one of those. If you're thinking, oh man, there's something I really need to focus on and get a sequence for, I need to know this, and I just don't know what to do with my afternoon today, I'd look at this and I'd say, okay, how can I understand moving from one molecule to the next? Now, with all that in mind, we're going to move through this. So we've got the first phase. The first phase is going to begin with glucose. So this molecule right here is our glucose molecule. We are going to invest 2 ATP in the first of our 2 phases. This phase is going to end with GAP or glyceraldehyde 3-phosphate. Our second phase is going to begin with GAP or glyceraldehyde 3-phosphate. It is going to end with 2 pyruvate molecules. We will invest 2 NAD+, 2 inorganic phosphate. From that, we will get 2 NADH. Ooh, neato. We will invest 2 ADP. Then we will return 2 ATP. And then again, 2 more ATP. This investment versus return phase and discussion is an important part of understanding glycolysis. There's a pair of very important words that are good for you to know when it comes to glycolysis. They are net and gross. So sometimes people will say, what is the net ATP generated from glycolysis? And the answer to that is 2. Think about that as what is the profit of ATP? Well, if you think about this like a business, you invest some money and then you get money back. What's your profit? Well, your profit is the amount of money that you get back after you've invested and you've accounted for what you invested. So we spent 2 ATP, but we get 4 ATP back. Well, our net ATP is therefore 2. Now, in addition to this, we're going to be introducing you, or I'm going to be introducing a couple of different enzymes. Every single enzyme catalyzed reaction, every single enzyme that is known, falls into one of 7 categories. I've got 6 listed here. I'm going to get the 7th one later this week. Every single enzyme is one of these 6 or the 7 later. They are either oxidoreductases, in which case the reaction utilizes oxidation reduction reactions. They are transferases, where they will transfer a functional group. They are hydrolases, where they will use water to break a bond. They are lyases, which will not use water but will break a bond. They are isomerases, which they will take a molecule, rearrange the atoms, and make a different isomer. Or they are ligases, where they will join 2 molecules together, making a covalent bond. So every single enzyme is going to fall into one of these 6 categories. And I bring these up because we're trying to learn glycolysis, and we're trying to learn glycolysis with respect to, well, it's a model, and all of the enzymes in glycolysis fall into one of these 6 categories, and all of the and 4 of these 6 categories are used in glycolysis. So the enzymes, hydrolases, and lyases are the ones that are not used in glycolysis. We're not going to lose a lot of sleep over these. Instead, what I want you to do is understand. Oh, just a second. What I want you guys to understand, oh, there we go. Okay, is the enzymes that kind of fit into one of those 6 different categories. So, with respect to glycolysis and kind of a summary of the overall reaction, we have our reactants, which are glucose shown here on the left. That is glucose. Pen. That's glucose. Okay, just a second. We have inorganic phosphate. Inorganic phosphate here. We have ADP, 2 molecules of that, 2 molecules of inorganic phosphate, and then up at the, we have NADH. These are all of our reactants. 2 inorganic phosphate, 2 NAD+, and 2 ADP molecules. Now, our products of glycolysis are 2 pyruvate molecules, shown here on the left, 2 ATP molecules, 2 NADH, 2 water, and 2 protons. One important key is I'm showing you the net result from all of this. So at this point in time, I want you to understand the starting point and the end point. This is the summary of all of it, not just the investment or just the return. This is the entirety of glycolysis. Now, here is all of that kind of summed up. Well, we're going to take 2 ADPs and we're going to phosphorylate them. When we phosphorylate 2 ADPs, we're going to end up with 2 ATPs. One of the things that's to me that's kind of cool about this is this molecule right here that I just circled, that's your glucose. That's nothing but carbons, hydrogens, and oxygens. And then this pyruvate, that's nothing but carbons, hydrogens, and oxygens. 3 carbons. You're going to take this molecule right here and make 2 of these. And in the process, you're going to make a series of high energy compounds, NADH and ATP. So essentially, you're going to be breaking some bonds of glucose very gradually, and in the process, making high energy compounds, transferring the energy from glucose into another compound. Now, what I want you guys to know about are steps of glycolysis. I can assure you that all the questions that you will have on exams will be something that you can answer if you can take the acronym for the enzyme and convert it to the long form name or the full name. If you can do that, then you're going to be just fine. Just a reminder, you guys have a note card that you can write on. So you can write on your note card and say, okay, well, there we go. I'm just going to write down glycolysis that way. That's fine. I have no qualms with that. That's space that you're using on that note card. Now, we're going to look at each one of these enzymes and each one of the steps of this reaction or these reactions and kind of go through them one by one. The first enzyme in glycolysis is an enzyme known as hexokinase. With hexokinase, let's take that name, hexo. Well, hexo indicates 6. Kinase. A kinase is a class or is a type of enzyme that transfers a phosphate from a high energy compound to another molecule. ATP is a high energy compound. Now, if you look at the equilibrium arrows here, what is that indicating? Tell me in the chat which direction these equilibrium arrows favor. Do they favor the product or the reactant, essentially? Forward. Absolutely. This is our first step in glycolysis, and this is an irreversible step. So our first step is irreversible. Okay. Now, with that in mind, one of the things that I want you to know is this molecule right here, glucose 6-phosphate, can go in this direction. It can go in this direction. It can go in this direction. And those different directions are indicating that this is a metabolite for a number of different processes. So what that means is that glucose 6-phosphate is not necessarily set on going through glycolysis. So we are not committing these atoms of glucose 6-phosphate. Yes, we have to go through glycolysis. Now, it can go through glycolysis, but it could also go to glycogen. It could also go to a pathway known as the pentose phosphate pathway, or as people know it, PPP, the pentose phosphate pathway. So glucose 6-phosphate can go in a number of different directions. This is our first step. It is irreversible. Hexokinase is an enzyme that transfers phosphates from ATP onto 6-carbon sugars. Now, what that means is that this enzyme has a lot of different sugars that it can bind to, but for our purposes, we're interested in its ability to phosphorylate glucose. Now, hexokinase is an example of a transferase. Now, this number right here, EC2, What that means is that, and all of these numbers basically give every enzyme a almost like a phone number, or if you're familiar at all with an IP address. This hexokinase has a number that is 2 point something point something point something. It's a series of 4 digits or 4 sets of numbers that specifically highlight, well, it is a transferase that uses a sugar as a substrate and transfers a phosphate group. And this is the exact enzyme that it is. So what I mean by that is every transferase, any and every enzyme that you find that is a transferase will have a numerical designation that starts with 2. Then we'll have another number, and then another number, and then another number. So these numbers, I want you to know this first character, the 1, 2, 3, 4, 5, 6, and what they relate to. So if I said, hey, I'm studying a new enzyme and its EC number starts with 2, I would want you to say, oh, well, that means it's a transferase. If I said, oh, its EC number starts with a 6, you'd say, oh, well, that means it's a ligase. That means you're joining 2 molecules and you're making a covalent bond. Now, this right here, phosphoglucose isomerase, PGI, phosphoglucose isomerase. We are taking glucose 6-phosphate, and we are rearranging it to make fructose 6-phosphate. Tell me in the chat what type of sugar glucose is. It might be easier to think about it. Oh, there we go. Never mind. Miracle said, oh, yeah, it might be easier for me to just know the answer. Done. I like it. Okay. Well, glucose is an aldose. What kind of sugar is fructose? Perfect. This enzyme essentially rearranges the atoms on an aldose to make a ketose. Okay. Now, looking at the numbers that we saw on the previous slide, what EC number would you assign to phosphoglucose isomerase? Perfect. It gets the designation of 5. 5 point. This would be 5 dot something dot something dot something. That 5 tells you it's an isomerase. Now, look at those equilibrium arrows. That's a reversible reaction. So our second reaction, our second enzyme in glycolysis is completely reversible. Just as you make fructose 6-phosphate, we can rearrange it and make glucose 6-phosphate. Favorable in either direction. Now, once we get this fructose 6-phosphate, from that we can make, well, there's our isomerization where we just rearranged atoms. We can now use phosphofructokinase, or PFK. Sometimes you'll see this written as phosphofructokinase 1 or PFK1. Now, with that knowledge, tell me about the equilibrium arrows for this and the reversibility of this reaction. Irreversible. Perfect. So our irreversible reactions are 1 and 3 are irreversible. 2 is reversible. What I said about reaction number 1 was I said that it was reversible. But I also said that glucose 6-phosphate could go in a couple of different directions. It could be used for glycogen. You could store glucose 6-phosphate in a glycogen chain. You could also metabolize it for the pentose phosphate pathway. then it could also go through glycolysis. PFK1 is an irreversible step, but in many cases, it is also known as the first committed step of glycolysis. What do you think that means with respect to fructose 1,6-bisphosphate? What do you think is going to happen to fructose 1,6-bisphosphate, the product of PFK1? Where's it going to go? Are we going to use that for glycogen? No. By this being the first committed step of glycolysis, this is going through glycolysis. Another way of putting that is that these carbons and all of the atoms that make up fructose 1,6-bisphosphate, their fate is to make pyruvate. Specifically, 2 molecules of pyruvate. Okay. Now, if you were to classify phosphofructokinase 1, how would you classify it with the EC numbering scheme? What number would you put in front of, or what number would be at the front? 2. It's a transferase. It's taking ATP, transferring a phosphate group onto carbon number 1. Here is carbon number 1. So carbon number 1 and 6 get phosphorylated. So now we have a molecule, fructose 1,6-bisphosphate. And to be honest with you, when you split it down the middle like that, it almost has a symmetrical appearance to it. What's on the left-hand side is, well, we've got 3 carbons and a phosphate, and then on the right-hand side, we've got 3 carbons and a phosphate. So it's an almost symmetrical molecule. It's not completely symmetrical, but almost. Now, that metabolite is a key metabolite in glycolysis. The next enzyme that is going to use that, fructose 1,6-bisphosphate on the far left here, is acted upon by aldolase. And what aldolase is going to do is it is going to split this 6-carbon molecule into 2 3-carbon molecules. These 2 3-carbon molecules have very similar appearances. A big difference, though, is that one of them ends up being an aldose. That's all. Here we've got an aldose. This is a ketose. Glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. Now, what I want you to notice about this, and this is something you'll probably do in metabolism, is I want you to notice this symbol is right here. And this symbol, this asterisk, is right here. This is used for carbon numbering. And the reason for this is when people study metabolism, one of the things that's very commonly done is to radio label carbons and radio label atoms. Use a C13 or C14 isotope of carbon so that then you can track it and say, oh, well, through this metabolic pathway, we get an even distribution of this and this. And then, oh, well, it goes into this cell type or this cell type, or it doesn't go into this cell. But what I want to illustrate for you guys is to say that this is carbon 1, 2, and 3. So when we look at glyceraldehyde 3-phosphate, what is carbon number 1 was previously carbon number 4. Then when we look at ketose, or our ketose for dihydroxyacetone phosphate, what is carbon number 1 right here was carbon number 3. So that's how those two molecules kind of get split in half. Aldolase is the enzyme that does that. Now, this is an irreversible step. There are a total of this is a reversible step. There are a total of 3 irreversible steps: hexokinase, phosphofructokinase, and pyruvate kinase. We take care of the first 2 irreversible steps in our initial phase of glycolysis. Our very last step, pyruvate kinase, is our last irreversible step. Now, what we've got here is 2 3-carbon molecules. Fructose 1,6-bisphosphate gets split into an aldose and a ketose, a phosphorylated aldose and a phosphorylated ketose. The enzyme that does that is a lyase, EC4, which is why we see EC4 right here. And what I was talking about was identifying all the numbers. Well, these different numbers, 1, 2, and 13, all say something different about this enzyme. They say something about what type of bond is being broken, what are the functional groups that are made, what is the substrate that is acted on, and that's why we have all these different numbers, so we can classify this enzyme and differentiate it from other enzymes. Now, the resulting products, if you remember the end of the first phase of glycolysis, the investment phase, we wanted to get to 2 molecules of this, glyceraldehyde 3-phosphate. We wanted to get to 2 identical molecules of that. When aldolase catalyzes its reaction, we end up with 1 of those and 1 dihydroxyacetone phosphate. Well, one of the things that I like to think about is in glycolysis, we phosphorylate an aldose. Then we take that aldose and make it a ketose. So we have this aldose to ketose conversion. We've done that. We did that with phosphoglucose isomerase. We then end up with an aldose and a ketose, but we need to end up with 2 aldoses. So what do? We take the ketose and convert it to an aldose. So half of our fructose 1,6-bisphosphate product, so this glyceraldehyde 3-phosphate, I'm going to put a smiley face by it because that one's good. Those 3 carbons are good to go. Those 3 carbons are arguably ready to move through glycolysis. The dihydroxyacetone phosphate, well, we have to use an enzyme to convert it to this. The enzyme that does that is known as triose phosphate isomerase, or some people call it TPI, some people call it TIM. And all that we're doing is we're taking our ketose, our dihydroxyacetone phosphate, and making glyceraldehyde 3-phosphate. Now, this is this is a reversible reaction. And once we have done this, once we have completed this, we have 2 glyceraldehyde 3-phosphate molecules, and we are done. Triose phosphate isomerase, it gets the EC5 designation because it's an isomerase. And that brings us to this point right here. And I feel like that is as good a point to stop as any, because we've made it through the first half of glycolysis. The rest of glycolysis, this is probably this just reveals a bias that I have. But when it comes to the interesting things about glycolysis, it's the investment phase, and then it's like the last step. So if you're looking at this diagram and you're like, what are the interesting things about glycolysis and what are the questions that he's going to ask? They relate to this circle right here. It wasn't intended to get larger and larger. They relate to this circle right here. And then this circle right here. The second circle is considerably larger that I drew, but in terms of numbers of steps, I think it's a little bit simpler. I think it's also interesting because in that first phase, that's where you have two of your irreversible steps. You've got an isomerization, you've got transferases, you've got a lyase that breaks a bond. It also gets you all ready with your pair of glyceraldehyde 3-phosphate molecules. Once you have that, to get from this point to this point, so from this X to this X, this arrow that I just drew, to put it bluntly, it's kind of boring. It's not actually boring, but it's like the sugars don't change very much. 1,3-bisphosphoglycerate becomes 3-phosphoglycerate, becomes 2-phosphoglycerate. But this area right here, from my experience of teaching metabolism and taking metabolism, this is where people run into the most problems. Just because they kind of lose sight of like what's happening. What you're trying to do is just one, you're trying to regenerate ATP, but you're also trying to kind of rearrange phosphates so they're in the right place. But in terms of like priorities that I have for where I'm going to ask questions, my circles is my first, second, and my third priority. So, and I think that's primarily because the first circle that I have, that's where you're investing ATP. You've got your and you've got kind of the, you're working with your sugar, you're rearranging, you're breaking it into 2,3 carbon molecules. And then the very last step is your pyruvate kinase, where you're ending up with 2,3 carbon molecules. Okay, that's where I'm going to stop. I didn't intend to zoom in like that. I'm going to stop my recording. I'm going to say thank you guys very much for coming to class.